theme is medicinal plants, and I would like to call upon our keynote speaker today, Vaidya Jayagopal Parla garu from USA, Global ERP Opportunities for Ayush and Wellness Professionals. Please come to the stage, sir. Call upon to the stage our chair today, Dr. Vijay Ganesh Reddy, sir, to the stage, Principal, Anantalakshmi Ayurvedic College, Warangal. Okay. Next, I would call upon to the stage Dr. Datta Kumar, Ayurvedic Rheumatologist, Hyderabad. Please come to the stage, sir. Today's theme, the medicinal plants. Plants up to date, there are more than 100,000 of plants globally, are either undiscovered or their medicinal activities are not investigated and analyzed yet. It is predicted that plants and herbs are playing an essential role in the medicinal field, especially in the treatment of critical diseases such as cancer and many more chronic diseases, so that their medicinal efficacy should be tested in the present era. So on this important note, we are planned this session on medicinal plants, and today's keynote speaker is Dr. Vaidya Jayagopal Parla, and the chair, introduction of chair, Professor Dr. Vijay Ganesh Reddy, Principal in charge, Government Ayurvedic College, Warangal, Anantalakshmi Ayurvedic Government College, and he has done his BMS from Usmania University and MD from NTR University, PhD from NTR University, and he has presented lot of scientific papers and attended many seminars and lectures he has given. So, and our introduction of the co-chair is Dr. Datta Kumar, Ayurvedic rheumatologist. He is working with integration of allopathy and Ayurveda, and encouraging many of Ayurveda students to attend rheumatology conferences and all. He is taking us, he took us to the next level when we were studying. Thank you, sir. And he is BMS from BRK Government Ayurvedic College, PhD from Pune College, and he has done his PG Hospital Administration and clinical practice of 30 years, and member of Osteoarthritis Society of India, and winner of many awards and paper presentations, and he is doing playing a significant role in rheumatology. And introduction of plenary speaker, Professor Vaidya Jayagopal Parla from USA. He is a graduate from Bangalore University, and MD in Ayurvedic Medicine from Rajiv Gandhi University of Health Sciences, Bangalore, and Masters in Acupuncture and Oriental Medicine from the Southern California University on Health Sciences, Certified Yoga Therapist, Professor of Ayurvedic Medicine and a faculty member at the Southern California University of Health Sciences and the Japan School of Ayurveda, founder and director of Athreya Ayurvedic Integrative Health Center, California. And I heard that his ancestors were in Ayurvedic followers, and they impact lot of impact on him. So he is into Ayurveda now. Thank you, sir, all the way coming from the USA for this occasion. We are very glad to have you here, sir. So
I would like to thank Swasthya organizers, and especially Dr. Satya, who has invited me to be part of this great congress or convention. Can you hear me all right? Yeah? Okay, thank you. So you can say that Ayurveda is going to be in its glory in the next 15 to 20 years. You have to be really prepared as Ayurvedic practitioners. I don't want to be partial here. I know that all Ayush stakeholders are here. I don't want to discourage you, but I will try to include as much of Ayush speciality as possible with the main focus of Ayurveda in my talk. So first thing that I would like to do is I do this with my classes every time I teach. So we'll close our eyes and then I'll chant for you. How about that? Okay? Please close your eyes and then we're going to chant. Om Namami Dhanvantarim Adidevam Surasurair Vandita Padapadmam Loke Jararuk Bhayam Mrityunasham Dataramisham Vividaushadina Om Shanti Shanti Shanti. Thank you very much. Systems is to include it, what we call as entire or whole system approach that we have. That's what is being is not only in the US but also in the European Union as far as Japan. We find this whole system approach being very, very famous, getting famous. So what is this whole system, which is nothing but what Siddha medicine says, Ayurveda says, to some extent homeopathic medicine also includes that, which is having sattva, which is the mind, atma, that is the soul, and the sharira, the body. So this approach that human being is an expression not of just a physical entity, but he is an expression of a psychological, expressions of emotions, and a spiritual being is now being really explored in the Western world. Thanks to yoga, yoga has done a significant amount of impact on improving this knowledge among the Western, audience or population. Can I have my presentation ready? Can I have my presentation? PPT. No, to health. Approach to health is changing. In the past, they thought that anybody who is free from disease, right? Disease-free state is health. That was the most basic definition for health. Then it has evolved to mental well-being, emotional well-being, social well-being, economical well-being. So all of that now has come down to the core principle of all these Ayush systems that we have, that mind body cannot be separated and we are conscious beings. So that's where we are starting, which Ayurveda and Ayush systems have been valuing for thousands of years, right? So that's why I said this is the golden era for Ayurveda, yoga, and Ayush systems of medicine. So that is the uniqueness that we are finding. And as Ayurvedic young aspirants of Ayurveda or Ayush systems, you should really be prepared for this expectation. What is expected of us to come to the global requirement of, whole body or mind, body, and conscious based medicine. Okay? So, did they find it or no? Yeah? Thank you. Good. So, what is the three aspects that were told to me to talk about was, what is the education? Second one is the practice. The third one is research. Right? We'll go in three phases. That's what I'm going to do. So, first thing is, I want to thank the Ayush Ministry and Ayush Department has done a phenomenal job in doing the groundwork for bringing Ayurveda to the level, Ayurveda and Ayush systems. For example, it may be Siddha or Saurigpa. Any of these systems need validation. So the validation and then the process of connecting to the region, to the Next slide, please. Yeah, first one. So what I was saying before. So here is what Ayush has done. 5 countries. I'm talking in the last 7 or 8 years, there have been 16 countries that have come forward for medicinal system into their policies. So here are the nations, 25 of them. Go next one, please. And here are some countries that recognize Ayurveda. Of course, SAARC countries, which is, the Sri Lanka, Nepal. So these SAARC countries have, recognized Ayurveda. But you can see 5 countries in the Europe have recognized it as a part of, healthcare or health modality. Next one, please. So here is my disclosure, what I do, and there is no anything holding back here. I'm a founder at Atreya Ayurvedic Integrative Health Center, Vice President for Atreya Herbs, faculty, etc. So next one, please. So some facts why people are looking for Ayurveda and Ayush systems of medicine is here. So keep clicking. So first thing is, this is a report from Harvard Business Review. It says that US health isn't built for primary care. They're accepting that their primary care is failing, right? That is because one thing that I appreciate in these Western countries is that they are very brutal in their own introspection. They will see where they are failing. So here they are saying the main modality or the best care, right? But if they go with, let's say, rheumatoid arthritis or osteoarthritis, they don't know how to handle it because the cost of care keeps on increasing, right? That is one thing that they are unable to bear, right? The next one, please. So this one shows that even though America has the most advanced health system, right? They are still seeing lowering of life expectancy. You know what is life expectancy? What is life expectancy? How much a person lives, right? America in the last 8 years has come down by 2 years. It was 78, now it is 76. They are unable to explain why this is happening even though we have the best health care, right? So they are now puzzled. Next one, please. Germany is not left behind. Germany is saying that the health costs are also going up, right? Next one, please. Here is another fact that we all as Ayush members will be really, equipped to is world aging population. The birth rate is coming down, aging people or aging population is increasing. So that means we have to care for people who are older. Japan is the one country where it has highest amount of aged people and they cannot take care of them. They have worked their life, they have a lot of money in the health system, but there are no people to take care because there are no youngsters. So this is something Ayush can bring in. Next one, please. So South Korea has the world's lowest fertility rate, right? So these are not a simple problems, they are problems of a nation. And Ayurveda can bring a lot of depth to this, and also Ayush systems. Here you can see in Japan, the total healthcare is about $2 trillion, which is an increase of 4.6% from the previous year. So what is happening is in the world paradigm, health costs are going up. Do you get it? The health, everybody is, thinking how to cut down their health costs. Next one, please. So here is the US, scenario or US, healthcare. So, in the US health system, spending grew about 2.7%. Some of it is because of COVID, of course, but they are saying that this is even because of chronic, conditions that people have to face. There are a lot of chronic conditions. Obesity is rampant in America and also in other developed countries where the cost of maintaining health of obese people is very high, right? So look at that. Preventive care in $4.3 trillion that America spends, only 3.5% goes for preventive care. Can you imagine? They are spending $4.3 trillion for taking care of acute care. Like somebody goes with, appendicitis or liver failure, kidney failure. They can only take care of that. They cannot care for health. Right? So that is something that is very noticeable. And what are these money? Where is it spent? This preventive care. It is spent on contraceptives, immunization, and wellness. That's it. So Ayurveda and Ayush can bring in a lot of depth of knowledge to enrich people and then empower them. Next one, please. So this is a study that was done by Zimmelman. And Zimmelman, she did a study showing that this was published in the New England Journal of Medicine. So there they are showing that the healthcare by 2030 will be subpar quality and will be not of patient safety. So they are saying whatever type of health system that they have, it is going to gradually fail. It is going to be substandard. People will not get quality care. Right? Next one, please. Second thing that we see in the world health paradigm is the crisis of mental health. In the US, there is a major concern about how people are suffering from various mental health conditions. These mental health conditions include anxiety, depression, substance abuse, such things. I'll show you a graph in a minute. So it is saying that the United Nations has taken a lot of interest in studying mental health, and they are saying there is about 25 to 27% of increase in prevalence of depression and anxiety around the world. Right? So these are the facts that we need to keep in mind and think about how, as Ayush professionals, we can start bringing more depth to the Ayurveda, to the world health paradigm. Next one, please. So this is a worldwide graph of 14 countries which are developed or developing or underdeveloped. You can see across, this was done in 2017, mood disorders, substance abuse, depression and anxiety. Everything is on the rise. Right? Next one, please. Last part is the spiritual health. From 2013 to 2020, there were 600% increase in the spiritual research. Can you imagine? 600%, the highest amount of research papers that were produced were on spirituality. Right? Chanting Om. I was involved in a study that was done in the veteran affairs in the sense, like in the army bases. So they are using Om chanting for taking care of their PTSD, that is post-traumatic stress syndrome. These are something that people are becoming more and more open to spirituality. So there is 64.1% of Americans look for some kind of spiritual healing during their health crisis. Next one, please. Here is a study that is produced by Zimmerman. He says that as health professionals, US health system has to educate their healthcare providers with spiritual knowledge. And Ayurveda, Siddha, and Saurigpa, they all start with spirituality. In my practice, if somebody comes, the first thing I do is I chant with them. I chant for them. right? Sometimes they ask me, are you not going to chant today? Because there is a connection. That connection, we may be seeing no value in it, but the places where they have lost their spirituality long ago, now they are recognizing this is important. Right? So we need to be more confident and very, very positive in bringing that spiritually, boldly. Boldly we should bring spirituality in our healing. Next one, please. This is another example here. During the pandemic, not only for the patients, but also for healthcare professionals like nurses, they used a lot of spiritual healing for themselves. This is a survey that was done by, this is actually a study from Poland. Next one, please. So what can, with all this, being given as a background, so what can Ayurveda and Ayush systems can bring in? So here are some of the, things that we can bring in. One is, as I said, there is Sattva, Atma, Sharira, that is body, mind and soul approach is very, very important. We cannot shy away from it. We should not think it is unscientific. First of all, we get discouraged. If somebody is chanting, we are going to think, oh, that is not Ayurveda, or that is not, Siddha medicine, or no, that is to be done, because you have to bring Ayurveda as integrative as possible. Okay? So that second thing is personalized healthcare. So personalized healthcare is another big shift that is happening in the modern system as well, because they have been doing generalized healthcare. Right? Anybody goes with a headache, aspirin was the medicine. Now they cannot do it. They have to now study a person before they give the medicine. So that's why personalized care is very important. Health empowerment, that means people who have larger, communities and populations and cities, they are now looking at how they can empower people to take care of themselves. Like people who are obese, how to motivate them to take care of themselves instead of they depending on the health systems. Community-based care, lifestyle medicine, whole system approach, natural remedies, diet and nutritional focus, time-tested medicine. Ayurveda and other systems are time-tested. We need evidence, but you can go with confidence because they are time-tested medicine. Next one, please. So here is yoga and its power. Yoga's power is incredible now in the US. It is the fastest growing health exercise modality in the US population. You can see 50 people who are 50 and above, 14 million people have interest in doing yoga, and then you can see the above, it is much more higher in the female population than the male. Next one, please. So here are the what John Hopkins University of Medicine says about yoga on their website, on the university medical website, medicine university website. You have this 9 studied benefits of yoga. Yoga can do all of that. And we, as a population, we have forgotten how we can benefit from yoga. But if you are a healthcare professional from, either Ayush or Siddha or Ayurveda, you should know about yoga. It can bring a lot of depth and value to your practice. Next one, please. What is going to happen is, the population here is going to experience what is being experienced in the developed countries, because we are also going to see the metabolic syndrome unfold here. In the next 20 years, metabolic syndrome is going to unfold because of affluence as Indian population we are experiencing. Here, current status of Ayurvedic medicine in the US, it is considered as CAM modality, complementary and alternative modality. It is recognized by the National Institute for Complementary and Integrative Health. Next one, please. How do we practice? If I have to practice, everybody may be curious, how do I practice Ayurveda in the US, right? You need to know about what is called as Health Freedom Act, and through which you can practice. There are 14 states in which the Health Freedom Act has been, amended, and using that you can practice. So what are the US what do we have, or any other country, including European Union or in the US, what are the main things that we can do as Ayush professionals or Ayurvedic professionals? We can do education. That's how I went to US. I started teaching, and they invited me to teach at the university, and they gave me a job. So if you want to bring Ayurveda, or you want to go to West, sharpen your teaching skills. Okay? If you can sharpen your teaching skills, then you will become really good in delivering the ancient knowledge of not only Ayurveda but Siddha and Yoga as well. Second thing is clinical practice. Third thing is herb business. Fourth thing is the research. Those are the four things that I'm going to talk. Next one, please. So here are national organizations in the US. So we are a population of about 1,500 Ayurvedic practitioners in the US. So we have quite a lot of organizations. We have National Ayurvedic Medical Association, which is NAMA, which is the biggest organization. Second is Ayurvedic Accreditation Commission. And then the third one is Association of Ayurvedic Professionals of North America. So there are like number of them. And also smaller state-wise. So in US what happens is you have state recognitions and then you have national recognition. So there are national associations and there are state associations. For example, in California, there is also National Ayurvedic Medical Association. So there are two associations. One is state and, next one please. So once you graduate, there are schools where I teach, there are schools we graduate about 100 to 150 students a year, and they come out and practice. When they graduate, they associate with one of these state associations or to the national association for their accreditation. So these are the NAMA certified courses. One is it is 3-level course. Or 3 strata of education in the US. One is the counselor, who is just going to talk about Dinacharya, Rutucharya and Ahara, which is 600 hours of training. The second one is Ayurvedic practitioner, who does not only Dinacharya, Rutucharya, Ahara, diet plans and all that, but they also do some herbs, some rasayana and some body work, that is Panchakarma. Then there is Ayurvedic doctor, which is 4,000 hours. So you can see number of hours are straight at 600, 1,500 and 4,500. Apart from that, there is also Ayurvedic therapist who gets about 1,100 hours of training. When they get this training, they can register, and then based on the Health Freedom Act, they can practice. Next one, please. So small internship programs are also recognized. So there are like doctor certificate courses, there are masters in Ayurveda course, there are seminars in Ayurveda, professional workshops in Ayurveda, like CEUs, like what we conduct here, CEUs. They also do CEUs there too for for updating their knowledge. Next one, please. So as I said, this is what is called as Health Freedom Act. So Health Freedom Act is based on each state. There are 14 states. California is one, Oregon is another one, Florida is another one. So there are 14 states that implement this Health Freedom Act, and one can use this Health Freedom. What is this Health Freedom Act? There is a declaration, there is a consent form. One has to fill in all the information, and they can agree to take treatment from or consultation from XYZ, whoever is an Ayurvedic practitioner, and they can have the services availed. Thank you. Next one, please. So what is Ayurvedic practice include? Single and polyherbal formulations. We have about 80 single herbs, and then we have about 18 formulations. That's what we get to choose from. So if you come to practice Ayurveda in the US, you should know every single herb that is there in the Dravya Guna. Otherwise, you will not be able to practice so well. So what we do for our students, we make them chant or learn the shlokas for specific, dravyas. Like for example, Brahmi, Ashwagandha, or Guduchi. We tell them to by heart these shlokas. The courses are very vigorous, and they are not like just learning something and then getting out. They have to learn quite a bit of information, and then they learn Dravya Guna, Chakaya Chikitsa, all these various subjects in depth. So diet recommendations can be done, lifestyle suggestions can be done, yoga can be done, and then Panchakarma therapies can be done. Next one, please. So we did a small survey in the US based out of our university clinic, which is in Southern California University of Health Sciences, which is a 100 years old clinic where we have an Ayurvedic department. And then I have a small clinic in Long Beach in California. Between those two, we saw who are the people who are interested in Ayurveda. No, we want to find out. So when we started that, we took about 120 patients. Next one, please. And saw what are they coming for. Number one condition that they come for is the digestive diseases, because you know why? Every affluent society falls to the trap of eating unwanted food or large amounts of food, and then that is the starting of metabolic syndrome, and of course those are the diseases in which you can see. Then next is anxiety, third one is pain, fourth one is stress management, and so on. So next one, please. So in conclusion, what we said is about 31% of the patients came for digestive disorders, which is like acidity or having IBS or irritable bowel syndrome or inflammatory bowel diseases and such. Also weight is a major issue in the US. So you can also see that Ayurvedic people felt that the Ayurvedic care is much more comprehensive than going to their personal care practitioner or their primary care practitioners. Next one, please. Coming to the products, Ayurvedic products are becoming quite, famous in the US. To take some names, turmeric is everybody's medicine in US. Everybody takes turmeric in the form of capsule, or they will take it as curcumin, which is an extract of turmeric. Now, ashwagandha has taken US market by wave. Everybody wants to take ashwagandha. So here are some companies that manufacture in the US: Banyan Botanicals, Atraya Herbs, Kerala Ayurveda, Kottakkal USA, Ayurveda, then Garrison. These are the companies. Now if you look at, if you are venturing into Ayurvedic herbal product market, then you should know all the stringent rules that are applied by FDA for importing the herbs, like heavy metal test, microbial load, etc. Next one, please. Next one, please. Oh, sorry, previous one. Here is the way how Ayurveda is being integrated. Integrated in the sense, it is being integrated in Western medicine, or it is included in acupuncture. I myself have a degree in acupuncture, and then you combine two of them. You do acupuncture and Ayurveda. You do chiropractic and Ayurveda. You do Western medicine. Like somebody is having a liver failure, they will do Ayurvedic medicine and they will simultaneously do Western approach to it, including naturopathy. Next one, please. This is how my practice looks. This is like a clinical setup. And then you have treatment rooms, consultation room, and then people are very much accepting Ayurveda as a full care medical modality, and that is exciting for all of us, and we need to be really prepared for it. Next one, please. In terms of research, one thing that lacks for us is an attitude to do research. I don't know why we have that type of hesitation. Even when I was doing my MD studies, I was very reluctant to do research. That's because maybe we think Ayurveda doesn't need any validation. But world looks at it in a different way. We need validation. I stress upon doing evidence-based research. If you do case studies, if you do case series, if you do cohort studies, if you do surveys, all these need to be properly recorded, or data needs to be collected, and then we can all put together this effort to bring Ayurveda to the Ayurveda and Ayush systems to a recognition to the scale where modern systems accept it. Next one, please. So here are the challenges: lack of high quality studies, lack of funding. Some private fundings are available, but again, there is no major, the Indian government has now started doing multi-center studies that earlier we heard about. And then lack of motivation is a major thing, as I said. Next one, please. What are the final competencies that as Ayurvedic and Ayush professionals we need to have? We need to have teaching and language skills, research skills. We should have knowledge of technology, integrative care experience, Diet and how to integrate, how to use, how can I work with an allopathic practitioner, how can I work with an acupuncturist, that's what we need to think about. Diet and lifestyle structuring ability, then a conscious based approach, yeah, and then counseling, not canceling, counseling, and then behavioral health and yoga and Vedic philosophy. How to convey this to the Western mindset is what we need to learn about. Thank you so much.
Thank you, sir. Any questions from audience? Any questions? We will take in the last, sir. Thank you. Thank you, sir.
Thank you, everybody.
Please be seated. A lecture from Dr. Jayagopal. Now, the scientific papers we would like to call upon Dr. Dinesh Jagtap. The paper title is To Study the Effect of Anti-Tuberculosis Activity of Gloriosa superbia on the Survival of Mycobacterium. I invite Dr. Dinesh Jagtap to come on to the dais and finish your this thing in 10 minutes. Paul is in rush, he has to catch a flight. So, I like to call upon chair and co-chair Dr. Dattakumar sir and Vijay sir to come on to the dais to felicitate the speaker first.
Very good afternoon to all and respected panelists. Myself Dinesh Jagtap, working as a scientific officer under the Rajiv Gandhi Science and Technology Commission, Government of Maharashtra. It's a SNT Council for Maharashtra. The work which I am going to present is just to study the effect of anti-tuberculosis activity of Gloriosa superba on the survival of Mycobacterium tuberculosis by using a radioexperiment techniques. Next. So coming to the Gloriosa superba, it's a herbaceous perennial flowering plant, mainly contain colchicine and family Colchiaceae. It's an alkaline-rich plant found in Western Ghats and mostly in Kerala and Tamil Nadu, some part of the Maharashtra. The metallic and petroleum aqueous extracts of Gloriosa superba has antimicrobial, mutagenic and antifungal properties. Why only tuberculosis? Because as you know, it's a communicable disease, highly contagious disease, and its causative organism is Mycobacterium tuberculosis, which attacks mainly on lungs, but in some cases it affects all the part of the body like spine, brain and kidney. So if tuberculosis that doesn't respond to the standard drugs, it's called as drug resistance, and I am targeting the drug resistance. So it is reported in literature that plant derived compounds have antimicrobial activity against various species of microorganism. Next. Coming to the Gloriosa superba, as I mentioned, it's a Gloriosa superba having family Colchiaceae, and its common name is flame lily, or the creeping lily or glory lily. The plant part used is the rhizome, and it's a climber as you call. It's a leaf trip having tendrils and reaching up to 4 meters. Next. Prevalence, if you see the WHO reports, India has for 2020, it gives 210 per 1 lakh population. It is estimated that almost 40% of the Indian population is affected with tuberculosis, and that's majority to whom have latent TB. It's another serious problem. In world rank, India ranks almost 36 position in incidences of tuberculosis. The global tuberculosis report of WHO is stank reminder that tuberculosis is now killing nearly twice as many people as COVID-19 every day. So it's a serious globally, not only for India, but it's a globally problem. And in 2021, it was estimated that 10.6 million people fell ill due to TB worldwide. Next. So purpose of the study, the study that it's a, as that Gloriosa superba, we called it as a herbal medicine because it's a natural source. Its minute contribution may minimize the low burden, that overburden of the toxicity. The herbal medicine doesn't possess the drug resistance, and this is the main focus. And antibiotics mainly for nowadays, it's used as isoniazid, rifampicin, paracetamol, and ethambutol and streptomycin. But the treatment, the patient has to take it for 4 to 6 months. So it is dangerous to stop medication early or without medical advice. This can allow TB that is still allowed to become resistant to the drugs. To overcome all these problems, the extract of Gloriosa superba plant was evaluated for antimicrobial activity using radiorespirometry techniques against the mycobacterium tuberculosis. The radiorespirometry technique involves use of label 14 substrate and scintillation detectors to detect the radio level 14 CO2, which are taken as a growth index. Next. Yeah, the study, it's a simple study design. It's an experimental study, in vitro study, where we have used plant material, then using radiorespirometry technique, estimation of counts per minute, that is labeled CO2, and estimation of yield of population of TB bacilli. And next result, discussion and conclusion. Next. Yeah, aim is just to evaluate the antimicrobial activity of Gloriosa superba to get a new potent tuberculosis herbal drug. It established a in vitro model system using mycobacterium infected peripheral blood mononuclear cell in conjunction with radiorespirometry, which could be a highly predictive of the therapeutic outcome of the new tuberculosis regime for a new tuberculosis drugs. Next. Yeah, objectives are simple: to study the effect of methanolic extracts of Gloriosa superba on the survival of mycobacterium tuberculosis using radiorespirometry. Use of radiorespirometry in plant extract, isolation of peripheral blood mononuclear culture, infection of cultured PBMC with mycobacterium tuberculosis, and testing its viability mycobacterium tuberculosis bacilli in PBMC culture using radiorespirometry techniques. So you can see the Gloriosa, how the plant looks, and the plant part used is the Gloriosa superba rhizome. Next. Yeah, it's a simple method where radiorespirometry technique involves use of 14C labeled substrate and scintillation detectors to measure the radio labeled CO2 and which taken as a growth index. Next. So this as it is a tuberculosis, it's a highly contagious bacteria. So all practices with TB was done inside the biosafety cabinet, but class 2A. It's a simple flow chart where preparation of media growth, then the use of LJ medium buds, then inoculation of media with tuberculosis. Next step is to preparation of preparation of alkaline scintillation calculate impregnated paper, then assembly the radiorespirometry vials and radio labeled substrate. Then using plant extract, we have taken 2 parameters. First is simply for the radiorespirometry assay, and next is the preparation of PBMC cells to isolate the radiorespirometry assay. Next. Yeah, a radiorespirometry plant extract. Gloriosa superba plant material, that is rhizome part, was collected. Small pieces were introduced into a liquid nitrogen and powder was suspended in a methanol and extract prepared. Extract was applied to the radiorespirometry vial and lag time interval was determined. Positive vial received no drug and negative vial received no inoculum. The extract of Gloriosa superba was introduced into other 50 micrometer and 100 micrometer vials. Next. So these are the results. If you see the first graph, it shows the growth of mycobacterium tuberculosis in the presence of Gloriosa superba. The blue line shows the positive control, and gray and the orange, yellow line shows the gray is the Gloriosa superba concentration of 50%, 50 microliter, and the yellow line is Gloriosa of concentration of 100 microliters, where the orange line is of negative control, where there is no bacteria is added. And if you compare to the effect of Gloriosa superba on mycobacterium tuberculosis inside the PBMC cell, you see the completely inhibition as compared to the positive control. These are the counts against the days. So as the days are increasing, the growth becomes deplete. Next. So if you come to a discussion, so radiorespirometry is a rapid method for measuring the growth of fastidious microorganism like M. tuberculosis. Principle of this is simple. It relies on the metabolism of radio labeled carbon substrate to an easily detectable byproduct. And then now tuberculosis bacilli converts acetyl coenzyme into phosphate and then subsequently to coenzyme acetyl enzyme that enters into TCA cycle, giving out this carbon dioxide. Thus, when grown in a medium like containing carbon 14 acetate using it expected it respired out the labeled CO2. And detection signal introduced in radio labeled CO2 absorbed in alkali scintillation fluor ignition paper, and it's read by a scintillation counter. It's a liquid scintillation counter. Next. Yeah, we also gone to the kinetic study where it shows in conventional method the absolute number of bacilli is figured out after the doing dilution and plating. In radiorespirometry, the dimension microbial population is sensed from its metabolic activity, and the tangent was made on the growth curve to meet the x-axis. This gives the lag time of different inoculum. Thus, in simple way, initial amount of bacilli decrease, the lag time increase. Next. Conclusion. Radiorespirometry is a very sensitive method to detect the metabolically active mycobacteria. Estimation of lambda or the lag time is the essential parameter to compare the rate of the growth of the Mycobacterium tuberculosis. Mycobacterium tuberculosis is one of the major causative organism of respiratory disease, which leads to a permanent damage to the epithelial lungs linings. These rapid techniques detects metabolic activity and gives first-hand information about the effect of the methanomalonic extract of Gloriosa superba on metabolic status of the bacilli. The current study indicates that the antitubercular activity of plant Gloriosa superba inhibit the growth of Mycobacterium tuberculosis, but further the studies are required to probe the actual mechanism of the effect of the extract and the active compounds in the plants. Next. Thank you so much.
Completing in given time. Please be seated. I'll call upon Dr. Ushali Gugarati. Her paper title is Evaluation of Echinacea angustifolia mouthwash against pathogens causing pharyngitis. Rushali Gugarati. Come. And I request you to complete in time, 8 to 10 minutes, please. Thank you.
Good afternoon, everybody. Myself, Dr. Rushali Sudhir Gugrati. Currently, I am pursuing my Masters in Homeopathy at Father Muller Homeopathic Medical College, Mangalore. My study is in evaluating the antibacterial effect of Echinacea angustifolia mouthwash against pathogens causing pharyngitis. Introduction. Echinacea angustifolia is routinely used in homeopathy for the treatment of blood poisonings, upper respiratory tract infections, skin infections and septic condition. Echinacea angustifolia is reported to boost the immune system that aids the body to fight off the infection. Internal mode of administration of Echinacea is known to be effective, but the utility of Echinacea as a mouthwash is less explored. Hence took this study to explore the effect of Echinacea mouthwash in the patients of pharyngitis. The purpose of the study was to determine the antibacterial activity of Echinacea angustifolia using mouthwash by semi-qualitative analysis. The effectiveness of Echinacea in reducing the pain of pharyngitis patient by verbal analog pain scale. The study design was based on prospective sampling technique. Study method was on purposive sampling. Sample size was of 15 subjects. Study period was for 4 days. Inclusion and exclusion criteria. Inclusion criteria: patients with throat infection along with the pain in the age group of 18 to 15 years were selected. Patients with only upper respiratory tract infection without any complications were selected. Pain of 5 to 6 on the verbal analog pain scale. Exclusion criteria: patient already on other treatment for the same complaints. Patients with any other illnesses involving the respiratory tract. So, intervention by purposive sampling method, 15 subjects fulfilling the inclusion criteria were selected, and the swabs were collected for the culture, and they were cultured in meconium and blood agar. Then we prescribed Echinacea angustifolia mouthwash for 4 days, twice a day. The mouthwash was prepared by adding 10 to 15 drops of Echinacea angustifolia in half a cup of warm water. The subjects were asked to follow up after 4 days. Throat swabs were again collected to substantiate the changes before and after the growth of bacteria before and after the treatment by semi-qualitative analysis. Magnitude of pain due to pharyngitis was assessed using verbal analog pain scale to evaluate the effectiveness of mouthwash in reducing the pain before and after the treatment. So this is a device called MALDI, which abbreviates as matrix assisted laser depolarization and ionization, was used to identify the different strains of bacteria. MALDI is an imaging mass spectrometer, has unique advantage for analyzing tissue specimen in a unprecedented detail. So this device helps us to find out the different strains of bacteria found in the culture. So it is like it does the work in hardly few hours. So you'll get the result and you can know what kind of bacteria is present in the sample. Next. So data from the samples were subjected to PET test for the analyzing of the pain, and McNamara test was used for analyzing the efficacy of the drug in the growth of bacteria before and after the culture. The statistical test was executed under the expertise of expertise statistician by using open source software Jamovi. So these are the cultural plates. This is before and after. You can appreciate the growth. The colonies of bacteria is reduced in the after treatment. Next one. So here the growth is seen in all the four quadrants. Growth is completely seen, whereas after treatment you can find comparatively less growth of bacteria. So these are the strains of bacteria found in each patient. These are 6 patients and these are the group of bacteria found in each patient. So this +4 signifies that the growth was in all the 4 quadrants. Whereas after treatment you can see it's +1, +2 or +3. That signifies the growth was maximum in the 1 quadrant or 2 quadrants. And there was as well as the growth was in all the quadrants in few of the bacteria. These are the other 15 cases where these are the group of bacteria seen, and this is the growth before and after. So results, we found the evidence of antibacterial activity against few bacterial culture from the patients, though through semi-qualitative analysis. Those are as following: Streptococcus aureus, Streptococcus salivarius, Neisseria subflavescens, Klebsiella pneumonia, Streptococcus oralis. So these are the few of the bacteria which we found reduced like comparatively less growth before and after, but statistically analyzing using McNamara did not show any significant changes in the growth of bacteria of all the strains of bacteria. These were the few above bacteria, the growth was reduced, but not all the bacteria showed the same result. But we found that there was significant evidence of mouthwash reducing the pain due to pharyngitis. The calculated p-value was 0.001. So this is the graph which shows pain before and after the treatment. So conclusion, the study showed that Echinacea mouthwash having antibacterial property was not effective in reducing the growth of all the strains of bacteria, but it was observed to be effective in reducing the intensity of pain in pharyngitis. Thus I conclude that the aqueous solution of Echinacea augustifolia may be considered as a choice for reducing throat pain in bacterial pharyngitis. Acknowledgement, I thank Father Muller's Homeopathic Medical College for their logistic and financial support, and my guide Dr. Anita Lobo and co-guide Dr. Beena Anthony for their support and guidance throughout the study. Thank you.
Thank you, Dr. Rishali, for completing in time. Dr. Deepika Pandey, her paper title is Critical Analysis of Shukra Shodhana Mahakashaya for its pharmacological actions. Dr. Deepika Pandey.
Good afternoon, everyone. Respected faculty members and delegate present here. The topic of my talk is Critical Analysis of Shukra Shodhan Mahakashaya and its pharmacological actions. Introduction. Next. Introduction. The word Shukra is derived from the Sanskrit root word Shuch, which means purity, clear and bright. In Ayurveda, the Shukra Dhatu consists of 2 types, Sthanik Shukra and Sarvadehik Shukra. Sthanik Shukra is Garbhotpadankar and Sarvadehik Shukra is Harsh Priti Bal Pushtikar. Vajikaran. Vajikaran is a comprehensive branch of Ayurveda which deals with the overall sexual health of an individual, and Shukra Shodhan comes under this branch. Shukra Shodhan is not mere purification or ejaculation of semen, whereas it includes the increase in quality and purity of semen. Next. The purpose of the study. As per recent report of WHO, around 17.5% of adult population, roughly 1 in 6 worldwide, experience infertility, showing the urgent demand to increase access to affordable, high-quality fertility care for those in need. Ayurveda is given a class of herbs which are known to purify vitiated shukra, termed as Shukra Shodhan Dravya. These drugs are proposed to correct semen defect which are either physiological, example motility etc., or morphological, example teratospermia. In cellular level, they can also be used in treatment of dysplasia, that is abnormal cell growth, which can modify into cancers. Embryologically, Shukra Dhatu is generative tissue, not sperm alone. The generative tissue and organs arise from Shukra Dhatu kalas present all over the body. Next. Formation of sperm and ovum, which have the ability to give birth, is the most evolved function of generative tissue. The sperm and ovum contain the representative of all the body tissue and organs. The generative tissue in sex organ give rise to sperm and ova, while the generative tissue in other organ is responsible for generation of the same tissue and organs. The purpose of this study is to explain the concept of Shukra Shodhan and to describe the various pharmacological and therapeutic applications of individual constituent drugs of Shukra Shodhan Mahakashaya. Next. Now the Shukra Shodhan Mahakashaya. Kushtha, Elvaluk, Katfal, Samudrafen, Kadamba Niryas, Ikshu Kandekshu, Ikshurak, Vasuk Ushirani, Iti Dashemani, Shukra Shodhanani Bhavanti. As we have discussed, the concept of Shukra, the Sarvadaik Shukra and Sthanik Shukra, now we'll move forward to the individual constituent of Shukra Shodhan Mahakashaya and its relevant researches. Next. The first drug is Kusht, which is Sausoria lapa, family is Asteraceae. Sausoria lapa root aqueous extract significantly decrease sperm abnormalities, testicular tissue and DNA damage against ethifon-induced reproductive toxicity in male rats. The other drug is Elvaluk, which is Prunus avium, family is Rosaceae. Effect of alcohol extract of Prunus avium on in vitro sperm activation of human semen samples showed significant difference in percentage of sperm motility and percentage of normal sperm morphology. Next drug is Katfal, Myrica esculenta, family is Myricaceae. A preclinical study of acetone and acidic acetone extract of fruit showed anti-cancer potential. The other drug is Samudrafen, which is Sepia officinalis. The cytotoxic and proliferative effect of Samudrafen is studied, in which the cuttlefish bone powder promotes osteoblast proliferation and may be a potential material to increase the number of osteoblasts in bone defect in the oral cavity. Now the question arises कि why अस्थि को स्टडी में क्यों इंक्लूड किया गया, ये क्वेश्चन हमारे दिमाग में आता है. इसका रीज़न आचार्य चरक ने दिया है कि अस्थि में वायु की सुषिरता के कारण जो सुषिर उत्पन्न होते हैं उसमें मेद धातु का पूरण होता है. मेद धातु का स्नेहांश से मज्जा उत्पन्न होती है और मज्जा के स्नेहांश से शुक्र की उत्पत्ति होती है, विच शोज दैट अस्थि इज डायरेक्टली रिलेटेड टू शुक्र. Next drug is Kadamba Niryas, which is Anthocephalus indicus, family is Rubiaceae. Study of Anthocephalus kadamba leaf extract on hematological and biochemical parameters of albino rat showed significant increase in HB, RBC and PCV at various doses of leaf extract, might be due to enhanced erythropoietin synthesis. Next is Ikshurak, which is Saccharum officinarum. Saccharum officinarum molasses altered testicular and ependydymal integrity via lipid peroxidation, thus reducing sperm quality and androgen levels in male Wistar rats. This is very interesting to note that this study shows opposite result. We will discuss this later. Next, Kandekshu, which is Saccharum spontaneum, family is Poaceae. The rural people in Vellore district of Tamil Nadu and Andhra Pradesh use fresh juice of the stem of Saccharum spontaneum plant in treatment of mental illness and mental disturbances by the Vaidyars. Antipsychotic effect of Saccharum spontaneum were seen in few animal studies. Next drug is Ikshurak, ichthyorrhoea, which is Astracantha longifolia, family is Acanthaceae. Astracantha longifolia seed showed significant increase in the sperm count as well as fructose level of seminal vesicle on male rats. Next. Next drug is Vasuk, which is Osmanthus fragrans, family is Oleaceae. Potential anti-tumor effect of Osmanthus fragrans oil was seen in prostate, colorectal and gastric cancer. In traditional Chinese medicine, Osmanthus tea has been used as a herbal tea for the treatment of irregular menstruation. The extract of dried flowers showed neuroprotective, free radical scavenging, antioxidative effects in vitro studies. Next drug is Ushir, which is Vetiveria zizanoides, family is Poaceae. No clinical or preclinical studies were found to show the effect of Ushir in male or female infertility. Few ethnobotanical references were found regarding the use of Ushir root decoction by Santhal tribe of India in the management of sexual diseases. Next. To find the combined effect of Shukra Shodhan Mahakashaya, we found two studies, which were, the first one is in which 11 infertile male patients with oligospermia were treated with Shukra Shodhan Janan granules with, and Arand Mool Kwat in patient of oligospermia, in which the results were significantly increase in sperm count, semen volume and RLP and SLP motility. In next study, 30 infertile male patients with teratozoospermia were selected and Shukra Shodhak Ghanvati is given and the study showed that there was statistically significant difference in the abnormal head of the sperm, whereas abnormal neck and tail had no significant change. Next. The probable mode of action. According to Ras, 2 herbs have Madhur Ras act on Vata Shukra Dushti, 2 herbs have Madhur Tikta Ras act on Pitta Shukra Dushti, 4 herbs have Tikta Kashaya Ras act on Kapha Shukra Dushti. According to Gun, 7 drugs have Laghu Gun that helps to improve the Agni and hence improve the formation of Shuddha Shukra Dhatu. Virya, most of the drugs have Sheet Virya. The Stambhan property of Sheet Virya helps to increase the ejaculation time. According to Vipak, most of the drugs have Katu Vipak, which are known to have Shodhan properties. According to Dosh Karmita, Kusht, Elvaluk, Katfal, Kadamma Niryas helps in the management of Vata Shukra Dushti, Kandekshu, Ikshu and Ushir helps in the management of Pitta Shukra Dushti and Samudra Fen and Ikshurak help in the management of Kapha Shukra Dushti. Next. The discussion. Shukra Shodhan Dravyas are not merely semen purifiers, but they act on both Sthanik Shukra, which is responsible for reproduction, and Sarvadehik Shukra, which is responsible for Saumyata, Prasannata, Snigdhavarna, etc. Therefore, few drugs have Madhur Sheath properties, which will enhance production of Shuddha Shukra Dhatu. Since the 10 drugs mentioned in Shukra Shodhan Mahakashaya, all the rasas except Amla and Lavan, therefore it can be divided into drugs acting on Vata Shukra Dushti, Pitta Shukra Dushti and Kapha Shukra Dushti respectively. Along with that, these dravyas predominantly have Katu Vipak, which have Shodhan properties. Why there is less data on fertility action of these drugs? Because most of these drugs have Katu Vipak, which has Shukraha property. That's why in case of Ikshu, we have found the opposite results. Charaka has described Shukra Dhatu as Charato Vishwarupasya Rupa Dravyam Yaduchyate, in which Chakrapani had described that if these drugs act on Shukra Shodhak, it will also act as Artav Shodhak. But both Shukra and Artav have opposite properties. Therefore, we can see such varieties of Ras Panchak and Gun Virya Vipak in these drugs. Next. Observation. 4 out of 10 herbs, which is Kushtha, Elvalu, Kandekshu and Vasuk, are controversial in terms of botanical sources. As per author's knowledge, very few preclinical studies are done, while no randomized clinical control trial has been done till now. In Charak Chikitsa, Vajikaran Prakran, Acharya Charak has mentioned Ikshu, Kandekshu and Kokilaksha as a content in Bringhani Gutika. Otherwise, no direct reference regarding use of other drugs mentioned in Shukra Shodhan Mahakashaya is available in Chikitsa of Shukra Dushti. Inclusion of these drugs in specific Mahakashaya may be dependent on potent or quick action of those drugs on specific strotas or dhatu. Next. Conclusion. It is evident that Charak has mentioned a set of 10 drugs having Shukra Shodhan action, which can be compared to varied pharmacological activities. The modern day researchers lack clinical evidences regarding the action of drug on Shukravaha Srotas, that is genital or reproductive system. So integrative approach in assessment of efficacy of these drugs may prove beneficial in combating global health issues like infertility. Next, thank you.
And now I would like to call upon Dr. Yogita Dhiman. Her paper title is Comparative Phytochemical Analysis of 5 Varieties of Nirgundi, Vitex genus. Welcome, Dr.
Respected jury members and all the delegates present here, good afternoon to all of you. Myself Yogita, PG, second year scholar, Dravyagun Department, AIIA, New Delhi. I am standing before you to speak on the topic Comparative Phytochemical Analysis of 5 varieties of Nirgundi, Vitex genus, under the guidance of Professor Dr. Tanuja Manoj Nesri, ma'am, Director, AIIA, New Delhi. Introduction. Ayurveda has described many medicinal plants for therapeutic purpose, and many of these plants have multiple botanical sources. Nirgundi is one such plant of genus Vitex, known for its Shofhar anti-inflammatory, Kushtahar against various skin diseases, and Shulhar analgesic properties. As per API, Vitex nigando is botanical source of Nirgundi, but botanically we find various sources of Vitex genus, which includes Vitex nigando, Vitex agnus castus, Vitex obtusa, Vitex cannabifolia, etc. According to the contemporary researches, other species of Vitex genus have also been evaluated for their anti-inflammatory, antioxidant, analgesic and hepatoprotective activities. This study is aimed at phytochemical review and comparison of 5 species of Nirgundi so that all of them can be potentially used as Nirgundi or Nirgundi Bhed. Next. Purpose of the study is divided into 3 parts: morphological comparison, physico-chemical study and comparison, phytochemical analysis, which is further divided into 3 parts, that is phytochemistry, HPTLC study, FTIR study and comparison. All this is done to find out the similarities and differences between the available 5 species of the Vitex genus. Next. Study design. Plant collection of plant from natural habitat, authentication and preparation of herbarium samples. Physico-chemical study, loss on drying and ash values were done. Alcohol and water soluble extractives values were found. Phytochemical study, primary phytochemical analysis using chemical test was done. Sophisticated analysis includes HPTLC analysis and FTIR analysis. Next. This is the study flow chart. First is the picture shown for the collection from natural habitat. Next is preparation of aqueous extract for phytochemical analysis and aqueous extractive value. Next is preparation of methanolic extracts for the same purpose. This is a picture for phytochemical analysis using chemical test, and the next picture shows HPTLC analysis. Next is FTIR analysis. Next. These are the 5 species of Vitex genus which we collected from the natural habitat. First is Vitex nigando. It is seen as a shrub or a small tree. In this, we can see that the leaves of the Vitex nigando is trifoliate or pentafoliate, and the leaflets are lanceolate in shape. The central leaflet is comparably larger than the other leaflets. Margins are entire or crenate. The surface, the above surface is darker as compared to the under surface, and the leaves on crushing gives a typical smell. Next sample is of Vitex trifolia, in which we see 3 leaflets that are entire and with crenate margins. The upper surface is darker as compared to the lower surface, and the leaves are 3 to 8 centimeter wide. The inflorescence in trifolia species is bluish to purplish in color. Next is Vitex obtusa. the leaves are opposite. Here we found simple leaves, not compound, and the leaves are 3 to 8 centimeter wide. The flowers present in this species is white in color as compared to other species where we found bluish to purplish inflorescence. Next is Vitex cannabifolia, in which we see the five leaflets, and the inflorescence is comparably shorter than the Vitex agnus castus, which is 15 to 20 centimeter long in Vitex agnus castus, whereas in Vitex cannabifolia it is 5 to 7 centimeter long. And next, this is physicochemical parameters done as per the SOPs mentioned by API. The first is loss on drying. It is usually done to measure the moisture content present in the samples. The loss on drying in Vitex agnus castus is 0.05%, in cannabifolia it is 0.11%, in Nigundo it is 0.07%, in Vitex obtusa it is 1.3%, and in Vitex trifolia it is 0.07%, which are within the normal limits found in API. Next is ash value. It is done to measure the inorganic compound present in the sample. In Vitex agnus castus it is 7.51%, cannabifolia it is 7.28%, in Vitex nigundo it is 6.90%, in Vitex obtusa it is 6.33%, in Vitex trifolia it is 7.58%, and these all are also within the limits mentioned in the API. Next. This is the alcoholic extractive value. The sample and the alcoholic extractive value for agnus castus is 17.63%, in cannabifolia it is 16.34%, nigundo it is 18.93%, obtusa it is 17.92%, in trifolia it is 18.27%, and these are also under the limits of API, limits mentioned by API. Next is aqueous extractive value, and these are also under the limits of API. Next, this is phytochemical analysis, and we can see here that in methanolic extracts of leaves of Vitex species, alkaloid and flavonoids are present in all the samples, whereas tannins, proteins and saponins are absent, and in aqueous extracts of leaves of Vitex species, alkaloids, flavonoids and saponins are present in all the samples, except tannin is present in nigundo, not in the other species. Proteins are absent because proteins are usually absent in all the leaves of the various samples. Next, this is HPTLC analysis, and we found number of peaks observed in the 5 samples were 8 in agnus castus, 8 in cannabifolia, 10 in nigundo, 8 in obtusa, 10 in trifolia. This shows that various phytochemicals were present in various species, but the similarity found was the maximum RF. The maximum RF was 253 in agnus castus, 2.56 in cannabifolia, 2.56 in nigundo, 0.26 in obtusa, 0.265 in trifolia. And this RF is usually seen in the phytochemical nigundo side, which is usually present in nirgundi. So we can say that the nigundo side is present in all the samples. Next, this is FTIR analysis. The FTIR analysis shows the same functional group present in the samples, like amine, salt and fluorocompounder present in all the samples. Next, discussion. Botanical source of nirgundi leaves is taken as Vitex nigundo, as well as source of plant of Herenica is Vitex agnus castus as per API. There are around 250 species of genus Vitex, out of which around 161 species are found in India. About 18 species are known in cultivation. It is derived from the Latin veo, meaning to weave or to tie up. Various experimental studies have been done. Various experimental studies have been done. Various experimental studies have been done on different species of Vitex to evaluate for different pharmacological activities, but no studies are available regarding comparison of activities produced by various species of Vitex genus. From the physicochemical analysis, it is evident that there are considerable similarities in the properties of all the 5 species. Phytochemical studies, HPTLC analysis and FTIR analysis also revealed that there are significant similarities in phytochemical properties of all the 5 samples thus collected. Next. From this study, we can conclude that though there are morphological differences in above mentioned 5 species of genus Vitex, but there are significant physicochemical and phytochemical similarities among them. So this may be considered as a lead towards similar pharmacological actions which can be further proved clinically. Hence, this study can prove as a pioneer work in accepting various species of Vitex genus as Nirgundi. Next. Thank you.
And next presenter is Miss Sharon Arthur. Her paper title is In Vitro Antifungal Activity of Homeopathic Medicine, Arsenic Album, against Fusarium oxysporium. 10 minutes.
A very good afternoon to everyone. I am Dr. Sharon, PG Scholar from JSPS, Government Homeopathic Medical College, and I am here to present my topic, that is, in vitro antifungal activity of homeopathic medicine, arsenic album against the fungus, that is Fusarium oxysporum. Next. Next. So we all know agro-homeopathy is one of the emerging fields, especially in homeopathy. And there has been many clinical studies which actually say that homeopathic medicines can help in crop productivity, increasing the germination, and also fighting off, like the plants can even fight from the environmental stresses such as bacteria and fungus. So I have taken one such fungus which is called as Fusarium oxysporum. Now this Fusarium oxysporum fungus is present, it's like a tropical fungus which is actually present here in the tropical soil, present in the soil, and mainly affects plants such as banana, cotton, date plants, etc. You know, there has been a recent study which says that the emergence of this fungus, Fusarium, is on a very steady high. And because of the chemical pesticides which have been used now, like they're showing lot of resistance, especially to it, and leading to lot more economic losses. We know that pesticides are so harmful, how they really enter the food chain and they can damage the whole system. Hence, we need an alternative, a homeopathic, a natural alternative which can help in these conditions. Next slide. So, you can see what are the diseases which actually Fusarium causes. Two main, just to sum it off. First is the root rot disease and wilting. Now, root rot, what do you mean by root rot? See, as you can see, it's like rotting of the root. That's it, simple. Can I have the next slide, please? So, this is called as the wilting. Now, if you carefully observe what exactly happens, the water supply is first like cut off, the leaves will fall down, and later on you will have the blackish and yellowish discoloration coming up on the leaves. So because of this, see why you must be thinking why we have chosen arsenic album, homeopathic medicine. See, in homeopathy, what we do is we are going to match the symptom similarity. That is, for example, in a patient, these are the symptoms of the patient, these are the symptoms of the drug, match it. Actually, to be very honest, we tried many other specific medicines, but we couldn't get actually any much result. Then we thought, why not think the weakness, which is so characteristic symptom, which is seen in arsenic, and the necrosis, which is a hallmark again, and let's try it, and then we have the result. Can I have the next slide, please? So before actually continuing the study, the identification of fusarium fungus is very important. So as you can see, few spores which are especially present and the long structure. So this picture has been taken at Osmania University. So we have collaboration with them and we are conducting this study. Can we have the next slide? Okay. So the method what we are using is the disc diffusion assay method. Now, what everything is done in complete aseptic precautions. Now, as you can see, these are the Petri dishes which is there. Okay. After proper sterilizing, as you can see the pink color, that is the media. Okay. That is called as the Asthana and Hawker media. What is this media? It's like the food for the fungus that we are going to provide. So after pouring that, if you can see, there is a small test tube in ma'am's hand that contains the fungus. So she is going to pour it into that and just gently move around so that we have an equal growth, especially of the fungus. Can you have the next slide? Okay. So now if you can see, there is a small circular, especially that's a disc. The method we are using is the disc diffusion. So the small white colored disc, as you can see, that is called as the Wackman's paper. We have to take that paper, put it into our homeopathic medicine, which has been procured from Schwabe company, that is in the liquid form, so that it will absorb everything. And then we have to place it in the Petri dish, keeping certain distance with that. The next slide. Yeah. So now if you can see, and then we have to label them. Now if you see, we have taken arsenic 6 potency. We are trying in different potencies. 6, 30, 1x and C. Now what is the C? C is the control here. We have been trying to use control, that is the chemical control. Triazole fungicide. Because we wanted to have a comparative analysis of homeopathic and the chemical one. Now many homeopaths here would say, ki, why have gone so lower? 1x potency. This was specially procured from Vashisht Homeo store and we have used 6 and 30. As I said, we had tried with various other medications, higher potencies, if you say in homeopathic, where we couldn't get result. So we thought, let's go little lower because agro-homeopathy is one of the newer fields for us too. So this is the labeling which has been done. Now what you do is you're going to keep this at room temperature for around 3 to 4 days till you find the growth coming up. Next slide please. Okay, so these are the medicines which we are using and this is the control which have been already 6, 30 and 1x. This is the arsenic album medicine which we have used in homeopathy. The next slide please. So now this is the result. Now after 3 to 4 days, if you can really see, the white color, everything what you can find is the fungal growth. While if you see, the Wackman paper is there. You can see, find the central disc which is there. That's the Wackman's paper which is dipped. Now the pink color, as you can see, that is called as the zone of inhibition. Now what does the zone of inhibition means? It means that our medicine is inhibiting the growth of fungus in that area. That proves that arsenic album is very effective against this fungus. Now, if you see, and this area has to be measured in diameter. Can I just have the next slide? So we have to write the zone of, you have to measure this, how much that area is there. So as you can see, arsenic album 1x and control both are having 8 mm, while 6C is having 6 mm, 30 is having 7 mm. Can we have the back slide? Okay, so if you can see, the control and 1x both are on the higher side, that is 8 mm. This is 6 mm, that is 7 mm. But now, if you carefully observe, as you can really see, there is certain growth like white color which is especially present there. Now what does that really signify? That's the fungal growth there. So that shows ki this medicine is like a weak fungicide. So we have to appreciate the beauty of homeopathic medicine being in a dynamized form where you can hardly find the particles, especially in homeopathy, and you can find such a clear zone of inhibition which is especially present. So, and 1x also you can find some the growth of. So you have to appreciate and say, like we can think and we can say that yes, arsenic album 6 and arsenic albums 30 can be very effective against this fungus. Now this is like the result which we have got. What next? How we are going to give this medicine to the plants? Because finally what we are doing is agro-homeopathy. Homeopathy, yes, to patients we have found wonderful results. We know homeopathy is working very well in veterinary science also. So how we are going to apply or how you are going to give them? Hence, we require a collaboration of agriculturists, botanists, scientists, homeopaths, everyone, an integrated approach where we can do some field studies also. Because there has been few papers like this medicine we have to either we can spray the medicine directly on the plants and see the result. So a vast conducting field studies and also the higher potencies also. The work is in progress. We are conducting this on higher progress at Osmania University, collaborating our college, Homeopathic College and Osmania University. So finally I would like to say is, this is like these preclinical studies especially, they are like a small window to a very bright future ahead. Thank you, and I would just like to say thanks to thank you sir. So I would really like to thank our Vice Principal, Dr. Srinivas Reddy sir, who's been our guide and support in all, Dr. Shashikala ma'am from Osmania University, Dr. Shrujan from Osmania University, who have been tirelessly working and helping us homeopaths to do these preclinical studies and having a good future. Thank you.
Amazing, amazing, Sharon, for a vibrant presentation. Now I would like to call upon Dr. Sudhanshu Shekhar Moharana to present his paper. Title is Integration is the Essential Path to Achieve Synergistic Action of Medicines for Total Cure. Dr. Sudhanshu.
All the panelists, juries, colleagues and listeners. Here myself and my colleague Dr. Nayana going to present where integration is applicable, because I am telling the story of failures instead of success and how integration makes, because every physician has a lot of good success cases, but these are the failure cases where we integrate so that we achieve success. Next slide. So we integrate here with a nutritional therapy and yoga with homeopathy. That is synergistic study, which 1 + 1 is more than 2. So there is 3 types of study. One is in the integration, one is antagonist study, where neither this medicine acts nor that medicine acts. Both are cancel each other's action. Another is ceiling effect. The ceiling effect is when the one medicine, another medicine given, then it stops the action of that only. And synergistic medicines is, and in nutritional therapy and yoga, always synergistic effect is achieved. No antagonist, no ceiling effect. Next slide, please. Why we need the integration? So, if medicine is acting well by its own science, own system of medicine, no integration is needed. If it is acting partially, then you refer to higher center. Even higher center, if it is not success, then we need the integration. Next slide, please. Protocol. when a homeopathic is going to allopathic side, no, particularly there is no, they stop the medicines, homeopathy medicine generally, because homeopathy medicine do not have the withdrawal effect. But a case is coming from allopathy to homeopathy, then there is a withdrawal effect. So we cannot stop immediately. So steroids you cannot stop, anti-epileptic you cannot stop immediately due to high withdrawal effect. So we have to, with that medicine, you have to give our medicine and slowly taper off. That is the rules. But in some cases, particularly cytotoxic drugs and dangerous side effect medicine and allergic to the patient, the medicine, that we can stop. Next slide, please. Background of biomedical knowledge in nutrition integration. To have a integration in nutrition, you should have a biomedical knowledge. Otherwise, it is very difficult. Simply giving nutrition is not so easy. So, biomedical knowledge you can get from the one course is there, SWAYAM course. There is a nutraceuticals, functional food and nutraceuticals, and that is the only research papers. Chemical analysis of all nutritions and their research papers from onion, turmeric, so many others are what are the chemical content and how it physiological effect and how therapeutic effect. All are in scientific way that is given. Next slide. Before slide. So here 11 cases we have taken for the study. One is lactose intolerance in children and adult. Another is baby developing rash, diarrhea or asthmatic attack during weaning period. In spite of taking milk and milk product, child suffers from anemia, gluten enteropathy and nutritional solution. Next slide. Milk given to children along with food or after food or while going to sleep may lead to develop calcium deficiency diseases. Whole oilseed preparation frying is tastier and nutritious than oil fried food, vegetables. Nutritional solution to severe anemia during jaundice. Dry fish and excess meat intake is potential food for stomach and colon cancer. Lemon tea is better than tea prepared with milk. Next slide. Homeopathic medicine with proper nutrition supplement and yoga can do wonder. Modern medicine with nutritional therapy and yoga can do wonder. Next slide. Objective is to find solution to above 11 problems with biomedical analysis. And second objective is to find out integration mode for synergistic effect of medicine for total cure. Next slide. Next slide. Methodology. Biochemical and biomedical interpretation of 11 stated problem has been done. Nutritional analysis with active pharmaceutical ingredients and therapeutic action of utilizing nutrition with medicine has been narrated with cases. Next. Lactose intolerance. This lactose intolerance particularly found in children and adult. In children, it is a lactase enzyme deficiency for which for some days this is stopped generally, and then it is a limiting disorder. So after 4 or 5 days it starts lactase enzyme. That time there is a lactose free diet, lactose free milk is available in market. And in homeopathy we can have the medicines like ethujasinapium, natrum carb, so which helps us, but for that period you have to give the lactose free food from the market. Next. I already told. Next. So in particularly lactose intolerance found in adult, particularly South Asia like Sri Lanka, India, Pakistan. So here there is a lot of options is there. So there is a they can take instead of milk, cheese and some of the soya milk or coconut milk or this options. Then weaning period. This weaning period, actually the rule is when we are weaning, the child is weaning, first is donkey's milk should be given, which is similar to mother's milk. If donkey's milk not available, then goat milk. If goat milk not available, lastly cow milk. And cow milk rule is 50% dilution. If child is allergic to cow milk, then what to be done? Next slide. That is due to lactoglobulin. Alpha lactoglobulin found in cow milk and buffalo milk is the main causative factor for that allergy. So next slide. Already I have told you. Next slide. So for here I am giving one solution. One of the 6-month child, when child was 6 months, mother again became pregnant, so no mother's milk. So he now donkey's milk and goat milk not available. So what happened? He was given, he had lot of diarrhea was there, and so severe diarrhea